Okay, let's dive into this explainer and imagine shrinking down 100,000 times to take a fantastic microscopic voyage. I will be your lead biological investigator for this journey. We are going to take the pages of your textbook and transform them into an absolute map of an entirely hidden world.
Because think about it. If you zoomed into a leaf, or, even a piece of your own skin, you wouldn't just see green mush or a flat, boring surface. No way! You would find a bustling, incredibly active, microscopic city. Everything living around you, including yourself, is constructed from these tiny, perfectly organized metropolises.
We call this city the cell. First discovered way back in 1665 by Robert Hooke when he was looking at a slice of cork, the cell is the ultimate fundamental building block of life. The textbook diagrams we're going to look at today are our literal maps for this fantastic voyage. They show us how every single multicellular organism comes from just one single cell that divides and multiplies.
So, here is our mission briefing. We will cover 1. Gateway to the microworld, 2. The master map, 3. Organelle assembly line, 4. The ancient ancestors, and finally, 5. The cycle of life. Let's get moving.
Section 1. Gateway to the microworld.
All right, as we approach the city limits, we encounter the borders that protect the inside of the cell from the chaotic outside world. We first hit the plasma membrane. Now, don't think of this as just a static wall. It's a flexible, selectively permeable gatekeeper made of organic molecules called lipids and proteins. It uses processes like diffusion and osmosis to actively regulate exactly what gets to enter and exit. Like letting oxygen in and kicking carbon dioxide out.
But for plant, fungi, and bacteria cells, you'll notice an extra, super rigid outer shield called the cell wall, which is mainly composed of cellulose. And this brings us to a really crucial point, the hypotonic media rule. When you place a cell in a very dilute solution, water absolutely floods into it through osmosis. The cell swells up, building massive pressure, but this rigid cell wall exerts an equal pressure right back, which literally stops the cell from bursting like an overfilled water balloon. Section 2. The master map. Before we actually breach the gates and head deeper inside, we need to consult our master blueprints. I want you to explicitly look at figures 5.5 and 5.6 right there in your textbook. We really need to juxtapose the plant cell against the animal cell. Notice the rigid outer walls and those massive central storage sacs, the vacuoles, in the plant cell drawing. Contrast that with the animal cell, which completely lacks that rigid outer wall and relies entirely on its flexible plasma membrane boundary. Our map shows they share a lot of structures for sure, but these key differences completely dictate how they survive.
Section 3. Organelle assembly line.
Okay, we've passed the gates and are now floating in the cytoplasm, that fluid filling the inside of the membrane. Let's take a highly structured tour of the factory floor. Floating right near the center of our map, we find the nucleus. This is the double-layered command center of the entire city. It holds the chromatin milieu, which is this entangled mass of thread-like structures that organizes into chromosomes when the cell divides. This is where the DNA and genes live. It dictates exactly how the cell develops and directs every single chemical activity.
Now, watch how all of this builds into an actual, literal assembly line. Look at the complex passageways of the endoplasmic reticulum, or ER. The rough ER has these little studs called ribosomes that manufacture proteins, while the smooth ER is busy building fats and lipids. Once made, these products are shipped straight to the Golgi apparatus. See those stacks of membrane-bound vesicles? That's the Golgi, and its job is to modify, package, and dispatch those products to their final destinations.
Keep your hands inside the vehicle, folks, because we are passing the lysosomes. These are membrane-bound sacs filled with powerful digestive enzymes meant to clean up waste. But if the cell gets damaged, they can actually burst and digest the entire cell, which has earned them the pretty intense nickname suicide bags. Next up, you can practically feel the heat coming off the mitochondria. Notice the deeply folded inner membrane on your diagram? Those are the powerhouses of the cell, generating the vital energy currency known as ATP. If we glance back at our plant cell map, you'll notice the plastids. These are totally unique to plants. You've got the green chloroplasts, which are packed with chlorophyll and actively capture solar energy for photosynthesis. And that contrasts perfectly with the white or colorless leucoplasts, whose main job is just quietly storing away starches, oils, and protein granules.
And this brilliantly brings us to the central vacuole, To put this into perspective, in a mature plant cell, this single, massive storage sac can take up 50 to 90% of the cell's entire volume. That is huge. It's full of cell sap and maintains the turgidity, or stiffness, of the cell, while also storing crucial stuff like amino acids and sugars.
Section 4. The Ancient Ancestors.
Let's take a really quick detour from our highly developed eukaryotic city to look at Figure 5.4. This showcases a much more ancient, primitive architectural design. Notice how prokaryotic cells, like bacteria, are just vastly different. They are incredibly small, usually just 1 to 10 micrometers. Instead of a well-defined nucleus, they just have this poorly defined naked nucleoid region with a single chromosome. And crucially, they completely lack all those membrane-bound organelles we just toured. Seriously, everything we just saw in the eukaryotic cell represents an absolutely massive evolutionary upgrade.
Section 5. The Cycle of Life. As our fantastic microscopic tour nears its end, we have to ask, what is the ultimate purpose of all this bustling activity, energy generation, and protein packaging? Well, the city eventually divides. To sustain the organism, new cells must be formed. Through a process called mitosis, the mother cell splits into 2 identical daughter cells, which is exactly how your body grows and heals wounds. And then there's meiosis, where cells divide consecutively to form gametes with half the chromosomes, ensuring reproduction and the continuation of life itself.
All right, we have safely returned to normal size. Now here is your challenge. Grab your textbook, look at that blank practice diagram, and test yourself as a cellular detective. Can you spot the Golgi apparatus or the vacuole based strictly on the tour we just took? Remember, every single time you look in the mirror, you aren't just looking at one person. You are looking at a walking, talking universe made up of trillions of these invisible cities. Keep exploring.